The short interspersed elements, or signs, known as ALU elements, are present in more than 1 million copies in the human genome and may represent more than 10% of the human genome. These transposable elements first became incorporated into ancestral primate genomes. They are not present in non-primates. Various primate lineages have amplified them to varying extents. There are not as many in orangutans, for example, as in the higher apes. Many ALU insertions occurred in the ancestors of humans and chimps after this lineage diverged from that of gorillas, and so they are not present in gorillas but present in humans and chimps. And then thousands of new insertions are specific to humans and no other primates, while thousands of others are specific to chimps. Other classes of transposable elements have also been active in primate lineages, such as the L1 transposons and the SVA retrotransposons. Of the latter, there are more than 3,000 SVA retrotransposons in the human genome. Some subfamilies of this group duplicated and spread prior to the separation of the African ape lineages, while other subfamilies of the SVA retrotransposons are specific only to humans. There are other areas of non-coding DNA which can experience selection and which can affect phenotype. So there are endogenous viruses which have become incorporated into ancestral lineages at various times, which have introduced changes. Also, introns and the regulatory areas around genes have changed, and some of these can be shown to be different, say comparing human genes to chimp genes. One strategy for screening for important areas of the genome is to find areas which have undergone a more than usual amount of purifying selection, in which deleterious alleles are selected against. The overwhelming majority of the areas which have undergone purifying selection do not code for protein and are therefore called conserved non-coding elements, or CNEs. Some of these have undergone amplification at different points in ancestry, such as one group which became amplified in early mammals. One strategy for finding the areas of the genome most responsible for the difference between humans and chimps is to screen for those areas which have accumulated differences at a rate higher than expected. These could be abbreviated the HARs, the human accelerated regions. One such region, known as HAR1, is active in brain development and the formation of multiple cellular layers in the cerebral cortex. Another region, known as HAR2, may have had a role in the dexterity of the human thumb in human lineages. In conclusion, many of the regions of the genome which introduce change and promote evolution may not code for proteins, and the study of these non-coding regions has provided a number of insights into the evolution of genomes in general and the human genome.